Okay there. Hello, Epidemics. So you may have seen the announcement on Blackboard or in your email. I was kind of sick over the weekend and didn't get assignments posted, so I decided to give you guys a break. No assignments are due this week. If there's anything that's late, you're welcome to work on that. If you're all up to date, great. Maybe you can spend the time working on your project or on stuff for other classes. Doesn't matter. The project itself is due on Sunday, April 30th, unless you're going to do a presentation to the class, in which case it will be the following week, the first week there in May. Okay. So the last time we were talking about Unit 731, which was a Japanese biowarfare program trying to develop biological weapons that they could use on their enemy, the enemy's countries. And during the Second Sino-Japanese War, they actually were successful, if you want to call it that, deliberately causing the release of biological organisms into parts of China, possibly Mongolia and the USSR. The USSR is now they're probably talking about the area that is now known as Russia. And they were planning to attack the United States with biological weapons, but they basically lost the war before they were able to do that. So as far as I can tell, this has been the most, again, I hate to use the word successful, but the most effective use of biological weapons in terms of number of people killed. All of the previous attempts that I can see, you might have gotten some results, but usually not huge numbers of people. And this particular unit out of Japan probably did cause the death of between 250,000 and 500,000 people. So that's pretty significant. Now, I mentioned at the end of our discussion about Unit 731 that some members of this unit were captured by the United States government and given immunity in exchange for information about what they had done. So the US did have a biological weapons program. You may be asking yourself, do we still have a biological weapons program? Officially, no, but there's a lot of speculation about that. So the US biological weapons program, the official biological weapons program, began in 1943. So Japan was doing its work on Unit 731. Germany was doing work on biological warfare, and they had their own biological weapons programs. And so in the US, we worked on this during this time period. And it lasted until 1969. So it was 1943 to 1969. In 1969, President Nixon officially abandoned the program, closed it down, said we're not going to try and develop biological weapons. There have been 2 international treaties about biological weapons. In 1925, 1995, the Geneva Protocol, you've probably heard of the Geneva Convention, and in 1972, the Biological Weapons Convention, both developed treaties outlawing biological warfare. The US only signed on to these in 1975. So kind of late getting with the international program saying that biological weapons should never be used. And officially, the US Biological Weapons Program, the research was being done for defensive purposes. The idea being we were studying how to make and process and develop biological weapons because we wanted to have defense against them if somebody else used them. Now, that's the official stand. However, the United States Biological Weapons Program was secret. This was not known by the general public. The information we have about this program is, there's not a lot of information we have, and some of it is probably wrong. Remember, people tend to rewrite history to make themselves look better. If you can get rid of documents showing what horrible things you did, nobody's going to remember that you did them. So the official policy in the US was deterrent. We're going to deter other countries from using biological weapons on us. We're going to figure out ways to defend ourselves against biological weapons. And we are going to retaliate if biological weapons are used on US troops. So that's our official stance. We do know that this research used human subjects, including military personnel. So people who signed up to join the US military and got biological weapons tested on them. We also know that there have been some releases of biological material on the public. We know of at least 2 large-scale tests which used a bacteria that is not a disease-causing, but was modified to behave kind of like anthrax. And when I say behave kind of like anthrax, I'm talking about movement of anthrax through the air. And these tests were conducted in San Francisco, and the New York City subway. Once again, we're kind of strangled by lack of information. Has the US ever used biological warfare? We don't know. We honestly do not know. There have been accusations that it was used during the Korean War. There were claims that the US infected animals, farm animals, specifically turkeys and pigs in Cuba, to kind of go after their food supply during the 60s and 70s. So there are claims, but we don't know the truth for sure. Either way. So that's biowarfare. Biowarfare is done by governments. What we're going to talk about next is bioterrorism. So biowarfare, governments, bioterrorism by individuals or non-government groups. So we're talking about things like Al-Qaeda. We're talking about individual people. This is what we're talking about. Most of the time, bioterrorism usually fails. I'm going to be blunt and call it failure. Few people are infected. If any. I mean, that's the way it goes. When we look at bioterrorism, there have been several bioterrorism attacks. So some of these cases involve HIV, trying to deliberately infect people with this virus. Remember, at the beginning years of AIDS, it was absolutely a death sentence. So if you got infected, you know, that clock was ticking on your life expectancy. Nowadays, we have lots of treatments. It is no longer a death sentence. People are living relatively normal, healthy lives with this disease. Still wouldn't want to get it, personally. More successfully, bacteria that cause food poisoning. It's pretty easy. Man, I can't type while I'm talking, can I? It's pretty easy to spread bacteria in public places, especially if we're thinking about food poisoning type bacteria and say a buffet. So if you go to a restaurant that has a buffet, they've got food out on warming tables. You just go and you get what you want. Yeah, that's a good target for bioterrorism. The good news is it almost never happens. So I'm going to talk about two of the largest acts of bioterrorism that have happened in the United States. So the first event happened in Oregon in the summer and fall of 1984, between August and October. So there was a small community called the Dales, and in this area, about 750 people got food poisoning, mostly with salmonella. Salmonella is a major cause of food poisoning. It's a naturally occurring bacteria that can very easily get into the food supply. 750, though, that's a lot. 45 were hospitalized. All survived. Okay. When they started doing these investigations in the Dales, trying to figure out where these people were getting salmonella, this is a pretty standard thing for public health to do. So public health says, look at this rash of food poisonings. We need to figure out where this started. Where did this come from? Where is the problem so we can fix it? And they do this by contacting the people who got sick. So they'll call you up. Hi, Miss Davis, I understand you got salmonella. I need you to tell me everything and everywhere you ate for the 7 to 10 days before you got sick. Okay, it doesn't always work out that easily. You can contact the people. They may or may not remember what they ate and where they ate in a given week. But enough people, especially when you've got 750 people, enough people will remember enough about their diet during that time period to start narrowing in on where this happened. And they narrowed it in to 4 different restaurants, specifically the salad bar on these 4 different restaurants. Now, when you have a salmonella outbreak, usually you can track it down either to one restaurant, or you can track it down to an item from the grocery store. Bagged lettuce, that is a big source of food poisoning. Flour, raw flour is a huge source of food poisoning. It's very hard to keep these kinds of bacteria out of bagged lettuce because it's already been chopped up, or from flour. So this was weird, right here, right across the top right here. 4 different restaurants. Local residents started pointing the finger at a local cult. The local cult was led by a man, Rajneesh. So Rajneesh was an Indian, native Indian, not native, US Indian, India Indian, who had moved to Oregon and started up this cult. So this is a picture of him right here. So what was going on? Why were the locals suspecting this? So there was a ranch near the Dales, and in 1981, several thousand followers of this Rajneesh moved onto this ranch. So there was this dramatic expansion of this cult, and there started to be land use conflicts. You add this many people to a community in a very short period of time, and you're going to have issues with the people who've already lived there. If we're talking about a small community, this is a big impact on that population. And so the people who had lived there, the people who were not members of this cult, did not like this sudden increase in population out on this ranch. And the local government, the county government, refused to issue more building permits. For the ranch. So why would they do this? Well, one of the things that you get when you add thousands of people is you get an impact on the sewer system, okay? If they're on the municipal sewer and water system, then you're going to worry about them using a lot of water and producing a lot of waste, more than the system was designed to support. If they're not on the community system, then we've got to deal with septic systems. And there's a limit the septic systems require a good piece of land. So you need to have a certain amount of land in order to have a functioning septic system. They would need to drill wells. Again, this is impacting the groundwater supply, which can impact the city water supply, especially in the western part of the US, like Oregon. Water is not as abundant in the western US as it is in, say, New England. So we've got these kinds of issues. If they're on city power or if they get hooked up to the local power system, can the power system support them? All of this kind of stuff going on. So the county government said, no more, you cannot build any more buildings. So the cult decided they wanted to add more buildings. They wanted to continue to expand. And so they decided to take control of the county government. And so they were trying to get on the county commission. They were trying to get individuals from their cult. One of them did get elected sheriff of the county, so in charge of the police, basically. And it wasn't that successful, although they did get the position of sheriff. But couple of the things they tried to do was they brought in more people, thousands of homeless people. So they would go to big urban centers, they would round up homeless people, bring them in, and try and get them registered to vote so they could vote for the cult. The county clerk basically said, not happening. And she enforced a regulation that prevented these people from being allowed to register to vote. Okay, so that didn't work. So what are we going to do next? About 12 people in the leadership of the cult decided to try bioterrorism. Now, they didn't want to kill people, but they wanted to make people too sick to go and vote. So the early attempts two county commissioners were made ill from glasses of water. So they put the bacteria in the water. These county commissioners drank it and got sick. They tried to contaminate produce at local grocery stores. Yeah, this didn't work. Yay. They also tried to put bacteria on surfaces and doorknobs at the county courthouse. That didn't work. So the next attempt were the salad bars. These were a test run. So they were trying to see if contaminating salad bars would work. So they tried these other things, didn't really work all that well. So we're going to try salad bars. They also had plans to contaminate water supply and to assassinate a United States attorney, as well as committing other crimes. Why they were going after a federal government attorney, I'm not sure. I suspect the court case went the wrong way for them. So this didn't really work all that well. The citizens of the county were suspicious of the cult. They knew or suspected that the cult were trying to rig the election and take control of the government. So the citizens of the county turned out in very large numbers to vote. So that really prevented the cult from winning very many positions at all. The ranch only had 239 people voted. That's not very many. They had thousands of people on this ranch. They had 7,000 or around 7,000 people on this ranch. So why did only 239 vote? Well, it turns out that the vast majority of people living on this ranch were not US citizens. So they had come in from other countries. All right. So the community knew that the cult was trying to take over the county government. They appointed investigators at the cult. The investigators from public health moved in, and they found a lab on the cult property. This is where the cult had been growing the bacteria. They had specialized equipment that is designed for growing large amounts of bacteria. They also had several different types of poisons and chemicals on site, and they had evidence that the group had experimented on people. So they had experimented on their own people. It's kind of a theme with terrorist groups and people willing to do biowarfare, isn't it? So the 12 conspirators fled. Several fled to Europe. The cult leader was arrested and deported from the US. He tried to go to a variety of different countries and was refused entry. So the only place that would accept him was his native India. So he had to go back to India. He resumed his teachings, and the group he formed in India is still in existence. He's been dead for over 30 years, but it still exists as a meditation retreat or resort. 2 of the cult leaders were convicted of attempted murder. This, remember, if we go back to the very beginning of this story, 751 sick people all survived. This is the most successful bioterrorism event on US soil ever. So that kind of gives you, I hope, an idea that bioterrorism requires a lot of resources and is very tricky to pull off, at least on a large scale.
What was the name of the cult?
I don't think I have that written down. I suspect I didn't write it down because the name was something in Indian that I probably can't pronounce. But if you look up terrorism and the city, the Dales, D-A-L-L-E-S, you can find lots of more information about it.
So like, wait, they never killed anyone though, right? It was just
Right,
that
nobody
they got people
died.
sick. No one died. Okay.
unless some members of the cult died in the experiments, but I don't know if that happened or not.
Okay.
Okay. So that is the largest bioterrorism attack on US soil. There was one other significant event in 2001, and that one involved anthrax. So 2001, in September through October, we all remember, well, okay, you may not remember this. Oh, yep. William put the name in the chat. No wonder I didn't write it down. Rajneesh Param, maybe?
Looks about right.
Okay. So when we think of 2001, we think about the World Trade Tower attacks. All right. Big, very deadly terrorism attack. But there was also a bioterrorism attack that was going on at around the same time, probably kind of inspired by the terrorist attacks on September 11th, 2001. And during this couple of months time period, a series of letters were mailed from Princeton, New Jersey. These were sent to places like the New York Post, which is a newspaper, NBC News, 2 Democratic senators, Senator Tom Daschle and Senator Patrick Leahy. Woo. All of the letters included photocopies of handwritten notes. So the original handwritten notes were not mailed. These were made these were photocopies. They used block text. Block text like this means that you usually cannot do handwriting comparisons. In fact, handwriting comparisons usually do not work very well. They don't have a very high success rate for being accurate. But regardless, block text makes it even harder. And the block test, this is, kind of classic for these. It would say things like death to America, Allah is great, and the date 9/11, death to Israel. These were really typical. These letters resulted in 22 infections and 5 deaths. Fewer infections, but a lot more deaths. Anthrax is a much deadlier infection than salmonella poisoning. Salmonella tends to infect more people naturally, so it's much more common of an infection than anthrax, but anthrax is more lethal. So in these 5 deaths, 2 of them were postal workers. So remember, these were mailed through the US Postal Service. 2 postal workers died. It wasn't always clear how individuals got infected, but when we look at the bacteria, it was all clearly from the same source. So bacteria, just like people, are going to have unique genetics per individuals. And when you grow large amounts of bacteria, they're going to have the genetic characteristics of the original spore, the original bacteria that was used to start that culture. So we can track it back and say, yes, this was all from the same source. Eventually, you know, if you do it for a couple of years, then it doesn't quite work that way. But in the short term, it certainly does. So as a result of this, we now have biohazard detection centers at major U.S. Postal Service distribution centers, and all mail going to federal government offices are subject to gamma radiation that will kill spores. So it will kill the anthrax. This is an anthrax wound. Okay. So why use anthrax? Anthrax is a bacteria. It has a case fatality rate between 20 and 80%. There's, like I said, there's genetic variability between different strains of anthrax. So there's differences in this case fatality rate. There's also differences on how it was contracted. So we talked about this with the plague, where we talked about how bubonic plague was least deadly, pulmonary and septicemic, more deadly, same kinds of things with anthrax. How do you get it? So the letters resulted in either skin or inhalation infection. Not surprisingly, the skin infection is the least deadly form of anthrax. It is very unpleasant. You get these large and painful necrotic ulcers. Necrotic means the tissue has died. That's what all this black stuff is. It is dead tissue. And they are painful. Recovery can take months. So it's a long, slow process. Inhalation form, it can take 2 months for symptoms to show up. So if you think about this, if it's discovered that you've gotten inhalation anthrax, it's not surprising that a lot of the cases could not be tracked back to a source. I'm sure you cannot remember everything you've done and every place you've been for the past 2 months. And a lot of these people were probably office workers that opened mail. And if you open mail for senators and you get something that looks like that letter in the previous one, yeah, you might just throw it away, right? Or put it in a file drawer for, oh, here's where the crazy mail goes, and just completely forget about it. So inhalation, typical flu or pneumonia symptoms, shortness of breath, chest pain, cough, but it is deadly and it can cause death within 48 hours of exposure, or symptom onset, I should say. So it doesn't, it gets in, it causes an infection. It takes a while to cause the infection, but once it does, it goes fast. So that's one reason that anthrax makes a good bioterrorism weapon. Anthrax occurs naturally in soil. In fact, it can infect grazing animals. So most cases of anthrax occur when people dig in infected soil or handle grazing animals. Cows, goats, sheep, they can get infected if you work with cows, goats, sheep, or their products, leather, food industry, then you can get exposed to it. There are around 2,000 cases annually worldwide. So it's not very common, but it's something that happens, mostly in people who work with animals and mostly outside the US. Fortunately, this is the negative for using anthrax as a bioweapon. Antibiotics reduce mortality. It's not a guarantee, but if you get it and you treat it early enough, antibiotics can really reduce that. And there is a vaccine. So the vaccine is not generally released to the public. And the reasons for this are a couple of different things. Military troops will get this vaccine, but it's a nasty and not very effective vaccine. So there are harsh side effects. People feel really rough after taking it, or as we put it, there's a high rate of adverse events, and it requires multiple doses and annual or yearly boosters. So that is why it's not commonly available. So those make it not as appealing, but it can be deadly. It can be found in soil. And the other big reason why it's an effective bioterrorism bacteria is that it forms spores. Spores are a dormant form of the bacteria. So not all bacteria can form spores. Anthrax can. And this allows anthrax to live in the soil for centuries. And it then it can start growing again if it gets consumed by an animal or gets on somebody's skin, or especially if it gets into a cut on your hands. So that makes it relatively easy to send through the mail. You don't have to worry about all the bacteria dying off before it gets to its destination. So this is the outside of one of those anthrax letters. You can see that the outside looks fairly innocuous or harmless. Its return address is a 4th grade classroom. Don't really expect to have difficult letters coming from a 4th grade classroom. So finding the source of the letter was very difficult. There were copycats. There were lots of copycats. And these could be mailed from anywhere in the country. And some had white powder in them. And that white powder was usually like baking powder, baking soda, flour, something that was not anthrax. Anthrax spores, when they're used for this kind of thing, are a white powder. When we looked at the bacteria, were from a strain known as the Ames strain. So we could identify the source of the bacteria. It was originally isolated from a cow in Texas in 1981. And it was distributed to at least 16 bio research labs, including USAMRIID. So USAMRIID, the United States Army Medical Research Institute of Infectious Diseases at Fort Detrick, Maryland. USAMRIID or Fort Detrick are the common ways this place is referred to. This facility has very high biosecurity facilities. So if you want to do research on Ebola, this is one of the few places that you can go and do this. This specific bacteria, not only was it the Ames strain, but it had several genetic mutations that led back to a specific class of bacteria at Fort Detrick. So literally they could say this bacteria originally came from this flask. That's some pretty good tracking back for a source. So we know where the bacteria came from originally. Okay, so we know that they were all mailed from New Jersey, Princeton, or the nearby area. We know the bacteria came from USAMRIID or Fort Detrick. So, who did it? So the investigations were primarily done by the FBI, the Federal Bureau of Investigation. They investigated a lot of people, including people who were involved with or linked to Al-Qaeda. And eventually they kind of narrowed in on a guy named Dr. Bruce Edwards Ivins. He was a researcher. He worked for 20 years on the anthrax vaccine program at USAMRIID. There was a rumor that the reason Dr. Ivins did this was because the Army was possibly planning to scale back research into a better anthrax vaccine. Okay, so that was supposedly his motivation. Dr. Ivins did have a history of mental health issues, and his therapists were concerned that he could become violent and homicidal. So they raided his house and found ammunition and bulletproof vests, or a bulletproof vest, but no weapons. So he had ammunition, but no guns. Okay, that was kind of strange. Dr. Ivins denied the claim. He said, no, absolutely not. This was not me. He eventually committed suicide. Possibly by overdosing on Tylenol PM. So you're like, Tylenol? Isn't Tylenol supposed to be really safe? Well, it's safe as long as you take the correct dose. But Tylenol can cause severe liver damage and liver failure, and you can take too much of it and end up dying. Now, I don't know that you can die very fast. I don't know what else is in the PM, Tylenol PM. But Tylenol itself can, like I said, cause liver failure. And there are some questions concerning his death. It's not clear if he did commit suicide, if he used Tylenol PM, or if he used something else. Again, the government, has put any documents related to his death, kind of behind closed doors. Now, here's the big problem with Dr. Bruce Ivins. He was not trained and probably did not have the skills to make anthrax into an inhalable powder. It's not an easy process. So could he do it? Could he make it inhalable? And if so, where did he do it? Nobody found a lab associated with him. And he would have to have a lab space. At least 419 people had access to this bacteria. At USAMRIID. Later reviews will say, later reviews by investigators who were not part of the FBI said that the FBI overstated their confidence in the genetic analysis linking the mailed strain of anthrax to this strain at USAMRIID. I mean, it was the Ames strain, yes, but was it the strain from one flask at USAMRIID? FDA may not have been completely accurate in how confident they were there. They could not place Ivins at the mailbox where the anthrax letters were originally mailed. They couldn't place him there. They found no spores in his house, which, if he was doing it in a makeshift lab somewhere, that would be pretty impressive if he didn't bring any home with him. The spores in the letter had been chemically treated, which was something that was not done at USAMRIID. And the bacteria there was not treated that way. The facility at USAMRIID was not set up for handling spores. They did not make spores on site. So where would Ivins have been able to practice or learn how to do this? If spores had been produced at USAMRIID, they could have spread through the lab spaces very easily. This was not the really high security locations that they were working with the anthrax. There was no evidence of spread in the lab and no one working there contracted anthrax. So is Dr. Ivins involved? I don't know. I don't think anybody knows. Okay. So this looks like a good spot to have you guys go off into your breakout groups. And you have two options. Let's give you options for what to talk about. You can either discuss how likely it was that Dr. Ivins was behind the anthrax scare, based on what I've told you here, or you're welcome to, Google the Wikipedia site on it. Or you can talk about the role of the US in doing biowarfare research. You can talk about the ethics of that, which hopefully we all agree it's not very ethical, but also the likelihood that it happened. So I'm going to give you 5 minutes in the breakout rooms. Hey, I'm getting ready to close the rooms now. 1 minute to wrap up your conversations. Okay, welcome back, guys. So what'd y'all discuss? What do you think?
One thing that my group discussed is that we thought it was like improbable that Dr. Ivan like did or manufactured the anthrax because he didn't have a lab. So like, how else is he gonna do it? Like go out and get it from the ground himself or something? But it just seemed very improbable on his part.
Yeah, that's kind of where I come down on the issue.
Like without a lab, how possible?
Yeah, they've never found the lab. What about the other group?
Our group talked more about the biological warfare likelihood, or not likelihood, but I suppose the likelihood that they were committing or not committing. Man, I cannot talk right now. Conducting, sorry, research into those fields. I think it's very likely, and I think even if it were definitely taking place, it wouldn't be common knowledge. It wouldn't be something they would openly admit to.
Yeah.
But even so, it says that they have the United States Biological Defense Program, which seeks to increase the nation's preparedness against chemical and biological threats through, improved threat awareness, advanced surveillance and detection, and responsive countermeasures.
Mm-hmm.
That would, categorically be defined as biological warfare. I suppose at least research, because how can you be prepared for something if you don't research it?
Yeah. Yep. I think we definitely should be prepared. Not sure what else, not sure if that's the only thing the government's doing. That's the tricky part. Okay. All right, so that's kind of the end of the conversation about biowarfare, bioterrorism. And since we've been talking about anthrax, which is bacterial, I figured the next good topic would be to spend a little more time talking about bacteria and talking about antibiotics, how those work, and the threat of antibiotic resistance. Okay, so bacteria. Basically, bacteria are prokaryotic, which is, you may have heard that word before, usually single-celled organisms. So prokaryotic describes the type of cell. It means the cell lacks a nucleus. So our cells are larger and they have a nucleus. The nucleus is where the genetic material is stored. And if you have a nucleus, you're called a eukaryotic organism. So bacteria cells are a lot smaller than our cells. They're much more simple than our cells. The vast majority of bacteria are harmless. There are bacteria all over your skin, all over your digestive tract. Pretty much every surface on the planet is going to have bacteria on it to some degree. And most of the time they don't do anything harmful. They break down waste products. They break down dead plant material. This is the kind of thing that they do. So they're not really infecting anybody. One of the other things that bacteria have that we do not, they have a cell wall. So our cells, we have a membrane around the outside. It's made of fats or lipids, and it does help to keep out excess water. It helps to keep out harmful bacteria. It keeps things inside the cell that the cell needs. The bacteria have a cell membrane as well, but they also have this cell wall, which is a harder, firmer structure around the outside. Harder is relative. So in bacteria, the cell wall is made of something called peptidoglycan. Woo! There's a big scientific word there. Peptide is what we call a small protein. So that's where the first part of the word comes from. Glycan refers to a carbohydrate. Proteins, carbohydrates, lipids, or fats are types of biological molecules. We hear about them in nutrition all the time. Are you getting enough protein in your diet, right? But we have them in our cells too. So peptidoglycan, it's a bunch of small proteins connected to carbohydrates. That's really all it is. They are supportive and protective. And why do we care about cell walls? Well, because the cell wall has a lot to do with what kind of antibiotic we use. It also provides a target for antibiotics. If you've got an antibiotic that damages the cell wall of a bacteria, our cells don't have cell walls. We're not likely to have really nasty side effects from that antibiotic, or at least not in our cells. So we tend to divide the world into gram-positive and gram-negative bacteria. This is biology. Biology doesn't fit into nice, neat little boxes. So not all bacteria fit into these two categories. But this makes a nice, simple division of most of the kinds of bacteria that infect us. And so there's the staining process called the gram stain that can be used to put pigment or color into bacteria so that they can be seen under the microscope. If the bacteria are not stained, you really can't see them under a microscope. So you stain them, and they look, the gram-positive and the gram-negative bacteria look different. So the reason for this is that gram-positive and gram-negative bacteria have differences in the outer surface of their cell. So we'll start down at the bottom here with gram-negative. They have an additional layer outside the cell wall. And this additional layer is another membrane. And membranes, we call membranes and cell walls different things because membranes are more flexible and membranes are generally made of lipids. So they have a thin wall, thin cell wall, and then they have a membrane outside the cell wall. And the lipid outside the cell wall is made out of lipopolysaccharides, or LPS. Okay, here's another big fancy science word. Lipo, there should only be one P there. Lipo refers to lipids, what we commonly call fats. Poly means many or lots. And saccharides refers to sugars. So we've got this extra membrane, and that lipopolysaccharide membrane makes it harder for things to get in or out of the cell. Okay. Gram-positive bacteria do not have that LPS membrane. And so the cell wall and the internal membrane are relatively permeable, meaning lots of things can get across that cell membrane. Okay, now, why do we care about gram-positive or gram-negative? Because two reasons. We care because some antibiotics can target that LPS membrane. Some antibiotics target the cell wall. So that's one. And the other is that whether they're gram-positive or gram-negative, determines if an antibiotic can get inside. Okay, so that's kind of ending on a science dense note, but we're going to pick up there next time, and we're going to talk more about antibiotics, and then we're going to talk about antibiotic resistance and why we should all be worried about it. Anybody have any questions on today's material? All right, in that case, I will see you guys on Thursday.
Thank you very much.
Thank you very much. Have
Bye,
a great day.
everyone.
Thank you.
Thank you.